All right, good morning, everyone. Glad to be here. I'm glad everybody could make it, wildlife enthusiasts. I'm pretty excited about sharing the wildlife at Pepperwood and what we're calling the hidden wildlife through our Wildlife Picture Index project. And also, I've kind of focused this so that this is a presentation through the lens of Stephen. So this is primarily how I see the preserve. And so a lot of the slides that you're going to see are things that observations that I've made, pictures that I've taken with my camera, and of course pictures taken with our Pepperwood cameras, as well as Pepperwood video cameras. All right, out on the trail one day. So when I was first asked to give this presentation, because the focus of my work is primarily with the wildlife cameras, I was really thinking four-legged fuzzy things. So I was out on the trail one day, heading to check a camera, and I came across this banana slug. So I stopped and knelt down and actually started having a little conversation with this critter. Turns out banana slugs are really good listeners. And I started talking to this banana slug saying, wow, you know, there's so much more to wildlife than just what I'm used to observing, and that this is really great that I ran across this banana slug, and then because what it did, it expanded my vision as to what I'm going to really talk about today, because there's so much more than the four-legged furry critters that I'm normally seeing. So I was out checking a camera one day, and out of the corner of my eye, I saw this very small thing rustling in the grass, and so I looked down and I picked it up, and it was this very tiny newt. And I kind of had the same response to this newt that I had with that banana slug. Like, what is your story? How did you get here? Where are you headed? Kind of having those type of things. But it also reminded me that I'd done a little bit of work with Pacific newts, and as a student at Sonoma State as a student at Sonoma State at Pepperwood, I did some research on some newts, and I learned that did you know there's 3 different kinds of newts at Pepperwood? And that this is an identification sheet that I made for our forest monitoring project. We're gonna be doing some forest thinning and some pre-sky burning, and we wanted to understand if the wildlife was affected by the work that we were doing, and it's a citizen science based project, and so we have volunteers flipping cover boards, looking underneath them, and of course newts is one of the things that they're going to see, and they really need to be able to identify them too. And but by making these sheets, I actually learned a bunch myself. I'm out on the preserve a lot, mostly on a 4-wheeler. I've really learned to slow down a bunch. My first couple of years at Pepperwood, I was really work-oriented, and I knew I had to get to my cameras and service them and get them done as quickly as possible. And after going out in the field with Megan Waller-Murphy, doing some wildlife tracking, I really slowed down. I really increased my sense of observation. And in doing so, I'm able to stop on the road prior to running over snakes like this. And so again, I stopped and had this little conversation with this snake, and it's just these amazing wildlife moments that I have. And did you know at Pepperwood we have 3 different kinds of garter snakes? So this is another sheet that I made. I also work in Healdsburg quite a bit. Audubon Canyon Ranch has Modini Preserve. I maintain some cameras up there, also a second project to the one that we have at Pepperwood. Here's a small rattlesnake that I encountered one morning. It was early, so this snake was coiled up, preserving what it had left. This was another encounter in Healdsburg. Of course, this was more during the day, and this snake was active. Things that I see quite often. So we have these hidden wildlife, but we have these wildlife that most everybody can see. Anybody that's out in nature that's making observations, these are things that we can see all the time if we take the time to slow down. This is a snake that I saw at Pepperwood. This one is kind of hard to see. It's a little bit in the grass there, but as you can see, its head is down here and its tail is here. Turns out this is a gopher snake. I really didn't know that much about snakes when I first got to Pepperwood. So pretty much everything I've learned about wildlife, I've learned at Pepperwood. And so I'm also a lifelong learner, a student. And it's good to be able to identify between a rattlesnake and a gopher snake. And here are two examples of the difference. Can't forget the aquatic animals. There's so many aquatic animals in our landscape. As you can see in this picture here, you can probably pick up right in the middle. There's a frog right here. So I got closer. Got a closer picture, and I was already familiar with this frog. I knew what it was because I had done a little bit of work with those at Pepperwood. But looking back to my identification sheets, I could see on the left that it was not a western toad, and it was too small to be a Sierra tree frog on the right. And because it was in running water, it was pretty unlikely that it was bullfrog, which are on the bottom. And so it was a foothill yellow-legged frog. Then we have this aquatic animal. As you can see, this one is still in the aquatic phases. It has gills. It still has its gills around its neck. And I knew what this was right away because I'd been looking at them previously. But most folks probably really haven't had a chance to see them. But this is a Pacific giant salamander in its aquatic stage. So here's a identification chart that I made for some salamanders, and the top left is an adult Pacific giant salamander, and there's some other ones on here. The one on the bottom right, the California slender salamander, that's a salamander that you'll likely see at Pepperwood. The one on the bottom left, the arboreal, and the speckled black salamander, those you don't see too much. And of course, there's the birds. I get a lot of bird pictures on our cameras, and it seems like only within the last couple of years I've really taken an interest in trying to learn more about birds and understand them. Here's a gallery of pictures of ravens. And I thought these were really neat. Ravens they have so much personality, and I really like the picture on the top right where it shows them taking off flight. Amazing. Here's a picture of two juvenile ravens that actually were born at the Dwight Center. We had a pair of ravens nesting up in our courtyard area, and these two were just out kind of hopping around, getting their wings ready. And there was four total chicks that turned juvenile. One of them actually had a wing injury, but we were able to get it to the bird rescue, and they rehabilitated it. And then they were able to bring that bird back out and release it at Pepperwood. So it turns out a happy story for these. So those were some of the things that you can normally see when you're out and about. So now we come into the things that you might sometimes see. Here's a picture of a beautiful gray fox. Here's a young bobcat. I've seen a couple of these at Pepperwood. Of course, we have our coyote. This picture actually used to be my screensaver on my laptop for quite a while. Of course, there's a bear there now, and I'll talk about that more in detail. So then we get into these ones that you're rarely going to see, the hidden wildlife of Pepperwood. The Occupancy mountain range is home to mountain lions. We do have mountain lions in our area. We've been seeing them on our cameras since we first set them up, 2012, actually 2010. Here's a little critter that most people aren't even aware exists. This is actually a spotted skunk. We see these occasionally, but we don't get very many detections of them. So when they do pop up, we're pretty excited to see them. This is a river otter that showed up right before the Kincaid fire, and here's another river otter. This one is actually in Healdsburg. Surprisingly enough, I saw we got this picture of this otter clear in the top of a watershed. So it was way up Pine Flat Road, way up at the top of the mountains, and not really an area where you would expect to see an otter. But because we had cameras there, and the cameras are working all the time, we were able to get this picture. Here it is up a little more close and personal. Another mountain lion picture. Pretty spectacular. And of course, the black bear. We have black bears in Sonoma County. We have black bears right here in the hills above Santa Rosa. This is a picture of a black bear in Healdsburg. Here's a picture of a black bear at Pepperwood. They do come in different colors. So sometimes a black bear will be, it'll be black in color, it could be brown in color, it could be cinnamon in color. So there's some variation. And of course, there's this little one. This is actually a post-Tubbs fire porcupine picture. Prior to this picture, we only had some nighttime pictures which are in black and white. So we were really excited when we got a series of porcupine pictures during the day. And as you can see, it's starting to green up a little bit. So this is just a couple of months after the Tubbs fire went through, 2017. And this is probably one of the rarest photos that we've gotten so far. This is actually a ringtail. We got this picture in Healdsburg at our grid there in 2019. You can see that this second picture is a little bit more clear, but it's definitely a ringtail. There are some other projects. There's some WPI projects in Napa that have got a ringtail. All right, I put this picture in there because this is actually to let me know that I'm going to be shifting gears here a little bit here. So now we're going to get into the wildlife monitoring at Pepperwood. There's a picture of a mountain lion, and if you look really close, you can see that one of its eyes is a little bit glassed over. So there's a good chance that this mountain lion is blind in one eye. So before we put up cameras, there's lots of other ways to see if there's animals out on the landscape, and that's going to be from your observations. We call that sign and scat. This top left is where you can see a picture of my hand next to a bear paw print. That's a front bear paw print. To the right of that, top right, is a scratch mark that you see in the soil, and mountain lions do that, and then they mark in that spot. And of course, the bottom left are claw marks, and the bottom right is a huge pile of bear scat. All right, so let's start talking about the wildlife cameras. This is really my main focus at Pepperwood. So what can you get from camera trapping? Well, certainly you're going to detect wildlife. We saw that. You're going to obtain some really interesting photos, and we've got to see some of those. But you can also determine presence and absence of species, of which I'm going to talk a little bit about when I start talking about the WPI. We can see habitat use, we can see different species in different vegetation communities, and we can definitely see some behavior. So without cameras, we wouldn't have got these pictures. We wouldn't have known in 2014 that we had a sow with a brand new cub. Assuming that this cub survived, it would have had its own cubs by now. So just a brief summary, just a little explanation of the Wildlife Picture Index. It's a study. With protocols that are established so you can set up a project, you can monitor that project, and you can do the analysis. And out of the backside of it, you'll get what's called an occupancy rating. And occupancy is basically the probability of detecting a species during a single survey given its present at the time. So really it's a presence and absence thing. And out of that, you can also, you can determine abundance, which is the percentage of composition. So like you can determine like if the system is balanced, if there's more of a certain species than of other ones. The picture on the bottom left is Sue Townsend. She's the one that brought the Wildlife Picture Index to Pepperwood. She used it in Mongolia, and then she brought it to Pepperwood as our principal investigator. And we were the first ones in North America to set up this kind of project. We're pretty happy about that. Here's a map. It's actually done, it's a modeling done by critical linkages. And this is just to illustrate when I start talking about corridors and cores and things like that. So the areas, the patches in green are what are considered core areas. Those are large intact pieces of land where mammals, wildlife can pretty much live all the time. They have the resources they need in those areas. So they don't necessarily have to go anywhere else unless they want to breed and spread their genetics to other populations. The yellow star that you see in the middle is actually where Pepperwood is located. These light green little trails that run between these dark areas are what are called corridors. Corridors are like wildlife highways. They're places that still have intact land enough that a large mammal can get from one area to the other. Quite often these are along creeks and riparian areas. So since Pepperwood set its grid up, there's been several others that have been set up. Pepperwood has two active WPI grids, and this yellow star on the bottom is Sonoma Mountain. We did a project there to determine how the effects of a new trail opening were on wildlife. The bottom blue star is in Marin, and that's One TAM, which is a collaborative of a bunch of different agencies. I think they have a little over 100 cameras there. And the three purple stars to the top right are projects that are in Napa County. So what are the components to the Wildlife Picture Index? So first of all, there's a part where you do your design, you put a grid, and then you lay that over a map of your study area, and you move it around so you can get as many camera locations on your property that you're working on, but also you want to have an equal representation of the vegetation community. So once you've established your design, which the cameras are a kilometer apart, you install your cameras, and then the cameras start taking pictures. In about 6 weeks, you can return, collect the data from those cameras, and then if the camera's knocked down for some reason, that quite happens quite often, things get broken off, cameras get defective, they get pushed on, they get moved out of alignment. That's what I do. I correct that when I go there. Then I bring the pictures back to the Dwight Center, the SD cards, and I download them onto our server where I batch name them and put them in folders and organize them so that we can get into the next process of information management. And that's where probably the majority of the time is spent on this project, the effort, and that is what we call cataloging. Cataloging is when we have to look at every picture and identify what's in that picture and then record that on an Excel spreadsheet. This takes a lot of time. And in the beginning, for the first few years, we had volunteers doing this, and they did an amazing job. And they cataloged thousands and thousands of pictures. After it's cataloged, it goes into what we call our quality control. That's where a specialist such as myself or somebody else at Pepperwood would have to go through and look at all these data sheets to make sure that the animals are identified correctly, that the count is correct, that there isn't any gaps in the data. Once that's all done, then it goes off into data analysis. This is where Dr. Morgan Gray, who's our environment analyst, she's now doing all of our analysis. And I'll be able to show you a little bit of the results from some of our early preliminary analysis. And then once that analysis is complete, then it goes off to decision makers for people like at the Sonoma Land Trust or county parks or private owners, so that people can make better decisions as far as wildlife are concerned on their property. A lot goes into this project. So before the cameras, you would have to have one biologist out taking observations. This costs a lot of money, and you can only be out there, for an 8-hour shift, and then have to come home. Or you can put a camera up in that same location, which can stay there all the time. Or you can do like we've done with the WPI and put 20 cameras out there. So it creates a lot more effort. It's a lot more affordable. It doesn't cost nearly as much as having 20 biologists out on the landscape 24/7, which just wouldn't be possible. So what is this WPI thing? Well, it's a long-term study, and in the very beginning, we established baseline occupancy estimates. So we determine who's there, and what time of the year are there, and what species are there. Over time, we can monitor trends in species, so we can see how the species are increasing or decreasing. We can see how animals are behaving, and also abundance. Abundance is the proportion of one species to the rest of the species in the total area. We also use that as occupancy estimates to assess the ecosystem health of the area. So if you look on the bottom center of this, you'll see a badger. We do have some pictures of badger, and I probably should have included that picture in some of those animals that we rarely see. Here's a map of Pepperwood with the green outline. We set this grid up in August of 2012. Like I said, the cameras are 1 kilometer apart. They actually spill onto our neighbor's property. Our neighbors gave us permission to set cameras up, so we're able to really get a comprehensive look of Pepperwood, but also kind of the areas around it. And I talked about a second grid, a second project. So we do have 2 WPI grids, one at Pepperwood, of course, and then the other one is at Audubon Canyon Ranch's Modini-Makima Preserve. Each project has 20 cameras. They're about 6.5 miles apart. So it requires a collaborative. We approached ACR and asked them if they would be interested in being a participant in the study, and they said yes. And so that grid, the second grid, was set up in 2013. Citizen science has been a huge part of the effort needed to do this project. In the beginning, it was a lot of field work was done by citizen science, and now primarily it's about citizen science cataloging. And more recently, we've developed an intern program. So where we have interns from Santa Rosa JC come and catalog the images. Also, we have Teen Nat alumni from Pepperwood who are also part of that program. After all that data has been analyzed, after it's been cataloged, it goes to the analyst. And right now we have a lot of analysis that are currently underway. Here's some pictures of some volunteers early on the project, all the way from when we first set up in 2012 to 2018. Here's a couple of the intern groups that we're cataloging. MESA students, like I said, MESA students, natural resource students from Santa Rosa JC, and also our Teen Nat alumni. Couple more groups, and then you can see the top right and the bottom left are their workstations in our library at the Dwight Center. So they come once a week for 6 hours, prior to COVID anyways, and they would get units for their work, but it was a paid internship. So we went from having volunteers do it to realizing that we really should be paying folks to do this, and it was just a great combination of having college students who get units and then they get paid, and that's been a really benefit to increasing the amount of images that we can catalog. All right, so what have we learned so far? Well, what we've learned is that it takes a lot of work to keep this project going. We used to, count our pictures, in thousands, and then we have over millions, and so now we're basically counting pictures in terabytes. Here's some early analysis that Dr. Sue Townsend did. So this is our first season, and basically this is what you would call your baseline. And we broke the animals into different groups. So you have your small mammals, your large ungulates, mesocarnivores, and large carnivores. So along the bottom axis, it would be your animals, and on the vertical axis on the left is your occupancy estimate. So I talked a little bit about occupancy, and that's basically the probability that you're going to make a detection of a particular species over a large area in a time period. The time periods that we use are 1 day. Here's some gray fox analysis. Year 1, year 2, broken down into seasons. So here's a coyote and opossum, year 1, year 2. So because this is so early in the study, you can't really make a whole lot of inferences by the data collected so far. This is primarily baseline data at this point. Here's some results from our black bear, year 1, year 2 at Pepperwood. As you can see, there was a gap during the winter. We're not really sure why that happened. Animals, black bears don't hibernate in California because we're in such a temperate climate. So here's a whole lot of information on one slide. So along the bottom axis, you can see the different species. In addition to individual species, you can see that each species has an estimate for one part of the season. So there's a summer, a fall, a winter, and a spring. As you can see, mule deer are detected almost all the time. So you can take that same graph and you can compare it to what we discovered at Modini Preserve in Healdsburg. As you can see, there's certainly some similarities going on here. So these are basically the baselines for the first year of each project, and it's actually the first year of Modini and the second year of Pepperwood. Really, the take home from this is that pretty much all of our species were detected. Here's a different way to look at the data. This is using pie charts. So the larger the circle, the more detections were made in that particular location. Each one of these locations is a camera location, but also it's broken into a pie with different slices. This is a better illustration of what each one of those circles looks like. So as you can see, the majority of animals are the mule deer, and then followed by the mesocarnivores, which takes a smaller percentage of the pie, and then the top predators, which even takes a smaller percentage of the pie. And that's really what you would expect to see. There's another one of my placeholders. All right, so we'll talk a little bit about the Tubbs fire and how wildlife responded to that fire. There are some fire pictures in here, and I know some folks could be sensitive to that. So I will let you know prior to fire pictures when they're going to start, and then I'll let you know when they're going to end if you choose not to view them. The little map on the bottom right shows the progression of the Tubbs fire as it moved across Pepperwood the first 3 days. There's a picture of a melted camera to the left of that. Remember that camera. Here is just a little a snapshot of what the pre and post fire detections were for the Tubbs fire. This is only a couple months before and a couple months after. So it's like I said, it's a really small snapshot, but it really kind of shows what you would expect to see, that there was a decline in wildlife after the fire. And more specifically, there was a 30% decline in carnivores and a 25% decline in herbivores. We don't have analysis beyond this point, so we can't really show you how wildlife rebounded from that statistically. So that melted camera is this camera right here. So this is 7 days before the initial firestorm, 1 day before the firestorm. This is where the fire pictures start. So if you choose not to look at them, I understand. I'll let you know when they're over. But the firestorm started on 10/9 at 1:57. And within a couple of minutes, it was totally engulfed. And then within 15 minutes, the fire had pretty much passed. So that's it for the fire pictures. So you can see it move through rather quickly. Well, there's another fire picture of it burning across the preserve later on. So there's 2 more slides of that. So it burned for 3 days across the preserve. So that's it for the fire pictures. So this is really what I wanted to get to. And so this is 1 day after the initial firestorm. And this picture is a little bit washed out, but that's because that camera was melted. But if you look to the right, you can see a buck. So 1 day after the fire, the buck are already returning to this area. 3 days after the fire, we see jackrabbits, which is pretty amazing. 4 days later, we see a mountain lion. 6 days, we see another deer. A week later, we see this mountain lion. 8 days later, we see a coyote. 12 days later, another buck. Again, 12 days, a different buck. 12 days later, another coyote. And still 12 days later, another buck. As you can see, there's still some hot spots. 2 weeks later, first responders are still mopping up. Big shout out to our first responders and the amazing work that they do to keep us safe. Thank you guys and gals. 2.5 weeks later, this is actually a part of the preserve that did not burn. So this would have been a safe refuge for animals to escape to during the fire. 2.5 weeks later, a coyote. A month later, a deer. Again, a month later, there's a bear in the background of that picture. Another jackrabbit a month later. And a bobcat. Squirrel a month and a half after the firestorm. Yes, we do have feral pigs at Pepperwood. And here is one a month and a half later. Another bear picture a month and a half later. A bobcat picture a month and a half later. And here's that porcupine I was telling you about. 3 months after the firestorm, we got the picture, a series of pictures of this, this porcupine. As you can see, there's starting a little bit of green up going on. All right, another placeholder. This was actually the very first bear picture we got at Pepperwood, and this was in 2010. So let's look a little bit look at the wildlife behavior. I have lots of pictures that show behavior, but But a couple of years ago, I set up some videos to see who was breaking my cameras off the stakes, who was abusing my cameras, because I wasn't quite sure who it was, not even knowing that I was going to get all these amazing videos. So here's a mountain lion sniffing the ground. Some mesocarnivores. Everybody's curious about my cameras. There's a rabbit doing what rabbits do when nobody's watching. Turns out squirrels are very photogenic. As are turkeys. And they're young. This is one of those forest monitoring plots where you can see that piece of wood in the background there. Here's a picture of a sow playing with her cub. Family of fox. Here's a buck chasing a doe. I think everybody knows what this is all about. I think this is one of the more interesting clips, the one of this coyote smelling this camera. I was often wondering why the teeth marks were on top of the wooden stake. As you can see, somebody pulled that camera over. It's about ready to fall onto the ground. Here's a picture of a gray fox moving one of its kits. This is certainly some hidden wildlife, something that you normally wouldn't see. Again, this is at one of our forest monitoring plots. So our volunteers lift up those cover boards and identify whatever's underneath them. Bear cubs are very curious. And a little bit mischievous. This is in September of this year. Oh, that's what happened to those cameras. That was in February. There's a couple of bucks wrestling. Behavior that you normally don't see or witness. Did you know that the black bears are omnivores? They eat vegetation, berries, nuts, grubs, and also meat. All right, so I've talked about a lot of stuff. Talked about the WPI, taking pictures of occupancy, of abundance, how important it is to have healthy ecosystems. So why is all that important? Well, it's important to understand what a trophic level looks like, what a balanced ecosystem looks like, and it's kind of a triangle with your primary producer on the bottom, and as you go up the ladder, you go into your primary consumers, your mesocarnivores, and then your top predators. There's always fewer top predators than there are primary consumers. So that first illustration was a little bit simplified. This is really how complex a food web is. So as you can see that they're so connected to each other that if you remove one or two of these species out of the food web, out of the food chain, things become unbalanced. So what happens if we don't assess ecosystem health? What happens if we don't establish functional cores and corridors in the region? What happens if we don't monitor trends in biodiversity, where abundance of key species, community composition? What if we don't take a look at the response to climate change and how we manage the land? Well, we start with what looks like a balanced system, but then if we remove that apex predator, then we've created an unbalance in the system. In this particular case, you'll end up with more ungulates, more deer. The deer will overgraze, and you'll have less vegetation, which really affects everybody else in the food web. So we looked at the animal part of it. Let's take a look at the land use part of it. Let's take a look at habitat. This is a model made by Adina Marylander for her study in 2010. And basically, it's called a connectivity report. And simply, the blue areas are patches of habitat that are still intact. So as you can see, we still have quite a bit of habitat. As the colors get lighter into the greens and the yellows, you start seeing what's called fragmentation. Fragmentation is where there's the lands are no longer connected on each side, and good possible chance that there's a barrier in the middle. So as you can see from top to bottom, there's this kind of a greenish yellow line that runs from all through this project. As most of you might have recognized, that's Highway 101. So already Highway 101 is a barrier to species on both sides. The yellow star is where Pepperwood is located. So you can see that below Pepperwood, there's another kind of a line that goes through there. That's Highway 12. So Highway 12, even though it's 2 lanes, is still becoming a little bit of fragmentation and a barrier to wildlife. If you look above the yellow star, you'll see that there's a line that comes from Highway 101 that kind of goes to the southeast all the way down into here. That's Highway 128. So at this point, we actually have still some connectivity, but if we continue to develop, if we continue to have land use changes, the more and more it becomes fragmented, the less ability the wildlife has to go from one population to another across these corridors and to spread their genetic diversity. So what happens if we don't do all the things and it gets worse? Well, we start seeing less and less of this. A healthy bear, sow with an offspring, animals breeding and reproducing and continuing on. Or we can look at the example of of the California tiger salamander. This is a salamander in its aquatic stage. You can see that it still has its gills. These are pond breeding species, particularly vernal pools. They only breed when it's raining. When the ponds are full, they have to grow up and leave before that. These animals are living in are using about 5% of their original habitat is left in the Santa Rosa Plain. 95% of their habitat is gone because of development, whether it be residential or commercial or agricultural. The land gets used up, the ponds get paved over or built over. So there really isn't a whole lot we can do with this particular species at this point because they're just barely hanging on. We can look at the grizzly bears. Grizzly bears have been extirpated from California for almost 100 years now. The only grizzly bear you'll see in California is on our state flag. This really wasn't so much as land development, but the inability for settlers moving in to live and coexist with this large predator. Native Americans were able to coexist for thousands of years with the grizzly bear, but as more people came in, the grizzly bears got pushed out and they're no longer in California. I can't talk about extinction unless I talk about the dinosaurs, of which, we didn't have really anything to do with that. It was other circumstances. But to leave on a happy note, I got to show you the picture of this dinosaur, that's alive and well, and out in the hills of Cazadero. I want to put a plug in for North Bay Bear Collaborative. Website is beingwithbears.org. This is a collaborative that's within its first year or so of development. Megan Waller-Murphy is the one that's driving this collaborative, but the Sonoma Land Trust, national parks, or state parks, local regional parks, Sonoma Ecology Center, Pepperwood, Audubon Canyon Ranch, we're all part of this collaborative. And what we're doing is we're really trying to be proactive to help people understand that there are bears in our neighborhood. They're right here in our hills. We've seen them come down into the cities, and that we can coexist with them if we behave in a certain way, if we treat our garbage in a certain way, if we just give them room. So this is something, if you're interested in bears, of course, as you can see, I am, because this whole presentation has been really bear heavy. But go to the website and check it out. There's some really interesting stuff going on. All right, we're getting near the end, so let's finish with one more video. This is a video taken by my friend Richard Vanlin in Healdsburg. The bear checks out the camera and then moves on.